A new CRISPR approach could treat hundreds of genetic diseases at the same time. And no, we're not kidding, this is real. The newest revolution in the field of medical CRISPR is here.
And of course, who but the lab of David Liu at the Broad Institute to bring us the freshest, most exciting new CRISPR technology.
As you know by now, David Liu's lab brought us base editing and prime editing, two technologies that revolutionize the way we do gene editing. Well, this time they might have outdone us.
They have developed a prime editing-based approach that could be directly used to treat hundreds or thousands of genetic mutations in different genes.
Now you must be thinking we hit our heads or something, but bear with us. We promise it will make sense. The key to understanding this are nonsense mutations.
Nonsense mutations are one of the meanest types of mutations because they insert a fake stop codon in the middle of gene sequence, kind of like planting a fake stop sign in the middle of a highway.
Nonsense mutations, which represent 1/4 of disease-causing mutations, block translation, resulting in a truncated protein that either gets degraded or can't perform its function. Very bad news for the cell.
Until now, scientists needed to develop a specific CRISPR approach to target each single mutation, like having to find and manually remove any wrong stop sign in the whole highway system. Lots of effort, money and time.
Here's where suppressor tRNAs come in. tRNAs are the molecules that load amino acids into a new protein during translation, and each tRNA binds a specific 3-nucleotide sequence, called a codon, and carries an amino acid.
And stop codons are recognized by special tRNAs, which instead of carrying an amino acid, stop translation.
However, it's possible to engineer different tRNAs that can overlook a stop codon and place an amino acid instead of stopping translation. These are called suppressor tRNAs.
Using suppressor tRNAs, scientists can coax the cell to ignore the fake stop signs in the middle of the road and continue its way to a fully formed protein.
However, until now, suppressor tRNAs had to be delivered externally, compromising their long-term expression in the cell.
To solve this issue, David Liu's team decided to use PrimeEdit to edit a redundant tRNA and make a new suppressor tRNA that is expressed directly from the cell's DNA.
And they engineered this suppressor tRNA to the last detail, ensuring that it's great at recognizing and skipping fake stop signs, but it still lets translation stop when it should.
How does that work? The same way the stop signs are usually accompanied by road lines and crossing, real stop codons usually have other sequences elements that help to stop translation, and the suppressor tRNA can discriminate those.
So, by installing this engineered suppressor tRNA, the team was able to instruct cells to ignore premature stop codons that cause nonsense mutations, restoring up to 70% of enzyme activity in human cell models without affecting normal protein translation.
And that's not all. They also applied this strategy, which they call PERT in vivo, in mouse models. They could see up to 25% restoration of full-length protein production. A fantastic result.
Using PERT, the team could rescue disease pathology in a mouse model of Hurler syndrome, a promising result for in vivo applications.
The most important thing is that they showed that the exact PERT approach can be applied to nonsense mutations in different genes. This is the first multi-gene therapeutic approach using CRISPR.
While the results are still preliminary, they are incredibly exciting. PERT technology could allow scientists to make one single therapy to treat many genetic diseases, something that until now was just a dream.
Of course, we are a long way from seeing this being applied to patients, but the signs point to a new revolution in the way we do CRISPR medicine.
We here with the CRISPR will follow this topic closely and be ready to tell you all about it.